I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. I'm your host, Jesse Lawler, excited to bring you episode number 65 of this podcast, dedicated to the improvement of your own brain by any and all means at your disposal. This week, first of all, I should make a disclaimer. This is water fast week. It's been almost 3 days since I've eaten any food at this point or drank anything except water. So if I sound slightly less perky than normal, please forgive the lack of glucose in my system. Now, I talked just a couple days ago with a real heavy hitter in the world of neuroscience, Dr. Trevor Robbins of Cambridge University. Dr. Robbins is the chair of the experimental psychology department at Cambridge and has a truckload of other credentials to his name. But what we're going to be talking about with him today mostly is something that he co-developed called CANTAB, which stands for the Cambridge Neuropsychological Testing Automated Battery, and is considered to be the world's most validated and sensitive touchscreen test for cognitive function. We're going to be talking about the development of that test over the last 30 years, which has obviously changed quite a bunch as technology has improved, and then some of the findings that have resulted from the use of that test on everything from monkeys to human beings, including not just the early prediction of cognitive disorders, but also which smart drugs seem to give cognitive benefits to healthy people. If you hang around until the end of the episode, in honor of Water Fast Week, I'm going to remind you of the various values of that can't say enough good things about it substance, water, and why you want to make sure that you keep enough of it in your diet. But first, let's do This Week in Neuroscience.
Smart Drug Smarts. This Week in Neuroscience.
So a recent study held at the University of Miami and led by neuroscientist Amishi Jha has lent further credibility to the benefits of mindfulness training, a form of meditation, in this case looking particularly at the ability of soldiers in combat zones and high-pressure situations to maintain their focus on the task at hand even when barraged by distractions and high-intensity stakes and all those things. There were 4 groups in this study, 3 of which were military personnel, 1 of which was civilian non-combatants. 2 of the 3 groups of military were given forms of mindfulness training. There was one that's called MMFT, mindfulness-based mind fitness training, and a second group was given an offshoot of MMFT that was primarily comprising of didactic information and discussions focused on stress and resilience. Then there was a military group that got no training, and the civilian group got no training as well. 8 months later, when the military personnel were deployed, they did follow-up tests, something called SART. SART is Sustained Attention to Response Task, which is designed to measure attentional lapses and mind wandering, which probably everybody wishes to avoid, but especially when people might be shooting guns at you. And it turned out that the soldiers who had received the MMFT training significantly outperformed both those soldiers who received no training and those soldiers who received the didactic discussion-based training. Interestingly, the civilians who were not in a high-stress combat environment also performed well on the SART test. So apparently less of a benefit to mindfulness training for those people that are already in comparatively low-stress environments. But, as the lead researcher pointed out, moment-to-moment information from the environment is necessary to ensure quick, decisive action. This is not just the case with soldiers, but also police officers, firefighters, trauma surgeons, day traders, pilots, and athletes, all of whom may benefit from short-term mindfulness training to curb attentional lapses and mind wandering. And chances are, if you're a Smart Drug Smarts listener, you're probably trying to be a high-performing individual, and that oftentimes comes with some stresses, so mindfulness training might really be something to look into. Mindfulness is defined as the ability to be aware and attentive of the present moment without emotional reactivity or volatility. And we'll probably have more on that in a future episode. In the audience interaction department, I wanted to put out a thank you to Smart Drug Smarts listener Stephanie D'Ambra for an email that she sent me sort of as a follow-up to last week's episode about the differences between the male and the female brain. A really interesting YouTube video. It's about an hour and a half long, videotaping of a course by Dr. Robert Sapolsky of Stanford on human sexual behavior. And he goes into a lot of the topics that would have fit right into our conversation last week about the many, many differences between male and female brains and how those morphological differences wind up expressing themselves as behavioral differences. So if last week just wasn't enough for you, we'll put a link to that YouTube video on the website. I also got sent to me a quick dictionary of surgical terms, since we were talking about snipping and cutting various normally thought to be important anatomical pieces last week. Here's 7 terms you might want to memorize, quick little suffixes. If something ends in tomy, it's the surgeon cut something. If it's an ectomy, the surgeon cut something out. If it's an ostomy, the surgeon made an opening. If it's a plasty, the surgeon changed the shape of something. If it's a pexy, the surgeon moved the organ to the correct place. If it's a raphy, the surgeon sewed something shut. And if it's a desis, the surgeon made 2 things stick together. So now impress your friends, amaze your enemies by making up strange surgeries that may or may not actually exist, but at least sound correct.
Smart Drug Smarts.
So Dr. Trevor Robbins is the head of the Department of Psychology at the University of Cambridge. He's also the chair of experimental psychology. He's also the director of the Cambridge Behavioral and Clinical Neuroscience Institute. He's a fellow of the Royal Society, the British Psychological Society, and the Academy of Medical Sciences. He was president of the European Behavioral Pharmacology Society, and he won that society's inaugural Distinguished Scientist Award in 2001. He's been included on a list of the 100 most cited neuroscientists by the Institute for Scientific Information, and so on, and so on, and so on. We are really lucky to have him here with us. And what you're about to hear is a fairly broad discussion on the development of the CANTAB battery. Again, that's the Cambridge Neuropsychological Testing Automated Battery. And then from the 30 years of CANTAB's history, what some of the highlight real moments are. Everything from Alzheimer's research to testing of cognitive enhancers. So with no further ado, Dr. Trevor Robbins. If you could maybe start off by telling us a little about CANTAB, the background of that program, and what it's looking to accomplish.
CANTAB is a battery of cognitive tests which gives you an assessment of your cognition. Cognition's many things, so it includes perception, attention, learning, memory, language, planning, and your ability to guide your behaviour and generally make decisions which are effective. It also stops you doing silly things like blurting out the wrong answer. So that's what CANTAB's about. It was constructed about 30 years ago, actually, and it works on a touch-sensitive screen. So the person works directly with the stimuli on the computer screen, which makes it easy. You cut out the middleman of the keyboard because, when you're typing on a keyboard and watching a screen, you have divided attention, and that's a problem for many people. If they have dementia or ADHD or something like that. So they interact directly with the stimuli, and you get very objective answers where you can also measure the accuracy and speed of their responding.
I didn't know that they had touch-sensitive screens 30 years ago.
Yeah, they did. They were great clunky things which you had to strap onto the computer separately, really heavy. And of course, eventually they evolved, as we all know, into the famous iPad. And of course, many of the CANTAB tests have also become available on the iPad.
And the CANTAB tests are unique in that they can be applied, like you have monkeys that do these same tests too. So it's really to give a broad spectrum of intelligence all the way from the animal kingdom up to humans.
The original idea about CANTAB is that it's translational. So all the rich evidence we've had from animal neuropsychology has been able to bring to bear into human neuropsychology. So all the systems, the cognitive systems that have been discovered in other animals seem to also work in humans for the most part. I mean, clearly there are big differences. Humans have language, for example, but the CANTAB tests are non-verbal. So they're based simply on visual stimuli, remembering and working with visual stimuli. For that reason, they're translational and can be used in other animals.
Can you talk some about the way that you've broken up the umbrella concept of cognition into some more granular subcomponents?
Yeah, sure. I think there's essentially three main components. There's what I call input systems, which is like perception and attention. Does the information get into the brain? There's then what I call representation in the brain. In other words, storing the information that you've input by learning, and memory, the code that you store the information in memory, which could be a language code or it could be a non-language code. And then finally, there's acting on those so-called representations to produce behavior, to sequence things in the right order and produce a plan, for example, or to make a decision based on reward. So those are the three main kind of categories of cognition that I would describe. Now, they probably correspond to systems in the brain. There's systems of the cortex which are specialized for perception, for attention, and for representation of information, and also for acting on information. An executive system, the frontal lobes of the executive system, which really coordinates all the other information and produces this effective plan or decision from it.
Thinking about the differences between humans and animals and how you could make a test that would work across the board for that, I think so much of our executive planning as humans, we use language for our internal representations of information. How the heck does an animal plan for the future when it doesn't have syntax?
Well, that's an interesting thing. I think, quite a lot of representation is language in humans, but we have a lot of non-language as well. Well, let me give you an example of a test which really shows this ability of the test to go across species. So I can show you an icon, a computer junk object, for example, which is quite attractive, and you can study it for a few seconds. And then at some point later, I'll show you that icon again, together with a novel icon you've never seen before. Now, a monkey or even a rat can distinguish whether they've seen that visual icon before. They will choose on the basis of reward which one they've seen before. And so can humans. Now, it may be that they use a different code to represent the icon. Maybe the human uses some language code and makes up a name for the icon. But the point is that the basic memory is visual. And so that is what's retrieved by the human and the animal in the retention test. And that test is called delayed matching to sample.
So over the course of the past 30 years, what have been some of the findings, I guess, that have resulted from having this common battery of tests that can be applied?
One of the real star discoveries has been this test called paired associate learning, where you have to learn the location of visual objects on the screen. So what happens is that imagine you've got 6 white boxes on the screen. They open up one by one to show a different visual icon hidden there. And you study this, and they open and close in a random order. And then after they've all opened and closed once, the icon appears in the middle of the screen. And your task is to put that icon in the white box where it was originally seen. And you have to do that for all of the icons. Now, that test turns out to be incredibly sensitive to detect Alzheimer's disease. So it probably can detect Alzheimer's disease. It's been shown at least 30 months or so before formal diagnosis. The idea of the test is simply that you're combining visual information with spatial information. That is one of the main functions of the hippocampus, of the brain. That function has been discovered largely by experiments in animals. Very different kinds of experiments, but basically the same kind of experiment, where, for example, a monkey may remember where an object is on the screen, which is to be rewarded. Or a rat will learn where a particular food flavour is in an arena. Or it will learn how to get out of a swimming pool where there's one platform which enables it to escape. So those kind of memories, you have to, in all cases, remember where the stimulus is, where the object or the icon is, and that's what the human has to remember. Now, the amazing thing about tests like matching to sample and this paired associate learning is that patients with Alzheimer's disease may do worse than animals. What you've got to remember in Alzheimer's is that you only get the diagnosis when you've been impaired in more than one domain of cognition. So remember those domains we were talking about earlier, like perception, language, memory, all of the executive function, frontal lobe function. Eventually, all of those things are hit in Alzheimer's disease. But you've got to have a hit in at least 2 of them before you get the diagnosis. Now, in fact, the memory change, the so-called episodic memory change, which the CANTAB PAL measures, that may occur by itself. And it may take a long time before other changes are recruited, which gives you the formal diagnosis. But the whole point about CANTAB PAL, for example, is if you can detect a deterioration in memory, and it's got to be a deterioration over a set period, let's say 2 years, then you've got, before the disease actually is diagnosed, then you could potentially arrest the disease with a drug. But you need to be able to detect that deterioration to test whether the drug is effective. But that is one of the success stories. Now, there are others as well. Just go in a completely different area. Modafinil. So it's a very controversial thing. You know, it's a very interesting drug. It's used clinically to treat narcolepsy. But interestingly, this drug, it's an atypical stimulant. So in other words, it's a bit like amphetamine, but it clearly doesn't work in exactly the same way as amphetamine. It's not as dangerous as amphetamine because it's not a drug of abuse. And this may be because when it's taken, it gets to the brain relatively slowly. And to be a good drug of abuse, or an effective drug of abuse, it has to get to the brain quickly. So modafinil isn't a drug of abuse, but it is nevertheless, it does have interesting properties. It improves some aspects of cognition. Now, in general, these aren't necessarily things like what we've just been describing, PAL. I don't think it's likely to be a really good treatment for Alzheimer's disease or something like that. But what modafinil is good at is probably improving your working memory and planning. In other words, your short-term memory, your ability to take on information quickly and form a plan. For example, to remember a phone number and then dial it. That's a very simple plan. And it helps maybe with cognitive flexibility a bit when you have to switch your strategies, as we were discussing earlier. So these are all kind of frontal lobe executive function. So we discovered, quite to our surprise, about 10 years ago, that healthy volunteer human subjects given modafinil showed improvements in some tests of cognition, notably planning, where they had to figure out a kind of chess-like problem, how many moves it would take you to get the goal. Now, of course, there's the whole issue about cognitive enhancers and whether they're used a lot by students and academics and others to improve cognition. So Barbara Sahakian, of course, has talked a lot about that. She published a big paper in Nature on this, and it's a real phenomenon. But the interesting thing is some of the work she's done in practical scenarios. So, for example, in a study with surgeons at Imperial College, apparently their boss was worried that they were drinking too much coffee to stay awake at night. And the problem with taking too much coffee is tremor, and you don't want tremor in a surgeon, do you? So they did a kind of lab trial of modafinil, and it improved their cognition, their flexibility, and reduced their tendency to make impulsive decisions, which is, again, what you want in a surgeon. When they were sleep deprived. So modafinil and shift work, for example, is an obvious application. In fact, the drug is used in America. It's often prescribed for shift workers to help them over the special problem. So there's always a place for drugs like this as long as they don't have long-term deleterious effects. And this drug's been around clinically for a long time. It doesn't seem to produce any major serious adverse effects. So presumably it's a good thing. Just to follow up on the CANTAB, in those tests, the CANTAB batteries, the planning test and the cognitive flexibility test, they're the ones that were shown to highlight these problems or to show the improvements anyway produced by modafinil.
So let's go back to cognitive enhancers for a moment. We talked a bit about modafinil. Are there other cognitive enhancing drugs or compounds that you've found good results from on the CANTAB tests?
Sure, yeah. I mean, the original one we worked with was Ritalin, or methylphenidate. This is the drug that's used a lot in the treatment of attention deficit hyperactivity disorder. And it produces, well, they say a calming effect, but it reduces impulsive behaviour and improves attention and working memory. And our original study, which really shocked us, was one we did in the 90s with Rebecca Elliott and Barbara Sahakian. We got volunteers, and these were Cambridge grads. We got volunteers to take tests from the CANTAB battery of working memory and planning, and we showed that even Cambridge grads got better the first time they had Ritalin. We thought, wow, so what's happening in ADHD? Maybe what these drugs are essentially doing is just boosting performance. So this is an interesting medical issue. So if it boosts performance in normal subjects, but maybe it's using the same mechanisms in ADHD. So methylphenidate was our first big example of a cognitive enhancer. And of course, that's why we went on to test modafinil, because we wanted to compare something else like methylphenidate to see whether you got similar effects. And modafinil actually produces effects which are quite similar to methylphenidate. Now, the other big class of drugs which are cognitive enhancers boost the cholinergic system, acetylcholine system in the brain. And we got to working on these very early on because in the 80s it was thought that loss of acetylcholine was the big thing in Alzheimer's disease. Certainly those cells do die quite early, and certainly if you give a drug to a volunteer which temporarily blocks the cholinergic system, you get all sorts of memory impairments which are reminiscent of Alzheimer's disease. And then on top of that, if you give a drug that boosts acetylcholine, you get an improvement. So we did a lot of work, actually, in the 80s and 90s with rodent models of the cholinergic system, where we showed that basically this system is very important for attention and for studying material, more than, say, just consolidating the material into memory. It's very good for the input and for encoding the material. And we found essentially the same thing in patients with Alzheimer's disease. So Barbara Sahakian did a big trial of one of the early drugs which boosted acetylcholine, an Institute of Psychiatry this was, with Raymond Levy, and showed that this drug, it's called Tacrin, did improve clinical symptoms of Alzheimer's disease, but the main effects you saw on objective tests were in terms of attention. So patients were better able to detect targets on a screen than normal. And we predicted this from the animal studies beforehand. Now, unfortunately, the drug didn't really improve objective measures of memory very much. And as we all know, that's the big problem. And the reason we think is because the network's already so far gone that the cholinergic drugs can't boost the networks. And, these drugs are still useful clinically, and a caregiver will certainly report that the patients seem to be, perked up, they're more attentive. Certainly they have a therapeutic effect. It's just symptomatic, and it doesn't arrest the decline that you're going to get. So the drugs are quite useful, but they're hardly very far from being a cure for this horrible disease. Now, another example of a cognitive enhancer is L-DOPA, because you've heard of Parkinson's disease, and that Parkinson's disease is a loss of dopamine in the brain. What people don't often realise is that there's more to Parkinson's disease than motor problems. There's a lot of cognitive problems as well. The kind of cognitive problems that are associated with the frontal lobes more than, like Alzheimer, memory changes. But there are cognitive problems. So what does L-DOPA do for them? Well, the interesting thing is, early on, we thought that they were also really good for the cognitive problems, and they are for some of them. But it turns out that this is one of these cases where there's a cost that goes with the benefit, because whereas things like working memory, remembering phone numbers and stuff like that, is improved with L-DOPA in Parkinson's patients, things like gambling, actually, risk-taking, is increased. So L-DOPA is a two-sided sword, as it were. We understand why that is in the brain in terms of costs and benefits. But it's another very interesting example. All of these drugs, acetylcholine and the L-DOPA, which affects dopamine, Ritalin affects dopamine and noradrenaline in the brain. All of these neurotransmitters are modulating the neural networks. They're kind of turning up the gain, increasing the signal to noise, improving the fidelity, in your recording. That's what they're all about. But there's another class of drugs which ambitiously would work within the network itself to turn on the switches and turn off the switches directly to really affect the computations that the network's doing, which underlie cognition. And these drugs, in theory, would work on glutamate, because the two major transmitters for the networks in the brain, one of them is a switch which turns things on, which is glutamate, and the other is a switch which turns things off, which is a chemical called GABA, or GABA-aminobutyric acid. Now, there are drugs which boost both of these agents. You can kind of modulate them a bit, but they're quite tricky and dangerous to use because, epilepsy, for example, can arise from too little GABA or too much glutamate. It can get out of hand, so you've got to be very careful with seizures and things. But there has been a theory that if you boost glutamate transmission, you might improve cognition. And this is the basis of the so-called ampakine concept of the early 2000s. Somebody called Gary Lynch, California, is a brilliant guy, suggested that, one of the receptors which binds glutamate is called AMPA. And if you can just make that glutamate bind more effectively to the receptor, it promotes, better firing, more efficient activation of the network. So he invented a few chemicals called ampakines. And, interestingly, in experimental models, in animal models, they seem to boost aspects of cognition, like, matching to sample, for example. And they even showed in one small study that it improved verbal recall in elderly normal volunteers whose memory was a bit shaky. But they never got anywhere further. And it's not quite clear why. There's often a lot of problems with these drugs, whether they really get into the brain properly, whether they have some unfortunate side effects, etc., etc. But for one reason or another, the ampakine concept hasn't won through in the end. But the fact that you can get these effects experimentally is encouraging. If you can, again, design your molecule to get rid of the side effects, then maybe you'd have an effective booster of cognition.
I'd love to actually jump back for a moment to ampakines. Why do you think, with some of these early findings showing reason for optimism, that the momentum there has been lost?
This is very interesting. Very often it's because you discover something else. There's some side effect. It may only happen in a few number of people, but nevertheless, that's enough for it not to be approved by the regulatory authorities. And maybe that's correct. So that's often the case. Another problem is that maybe you find in order for the drug to hit the target in the brain, you have to give so much of it that the dose is unrealistic and you can't really give the drug on that basis. For example, it may have to be injected rather than taken orally. And, there's all sorts of problems then which mean that you can't really use the drug in the way that you'd like effectively in a clinical setting. But, there's all sorts of reasons why drugs fail.
If you could do your dream research project at this point, what would it be?
This is a really interesting question. I've just got a dream project funded, actually, by the Wellcome Trust. I'm very happy. I'm very excited about it. I'm a senior investigator of the Wellcome Trust. And interestingly, what I'm working on mainly is OCD, obsessive compulsive disorder. We haven't even mentioned that yet, which is very poorly treated, by the way, and addiction, and what we call impulsive compulsive syndromes, these strange behavioral syndromes. And the reason I'm working on that is because we know a lot about the brain areas which control these behaviors. And I think we're going to have a big breakthrough in this area, either with behavioral therapies or with drug therapies or even with brain stimulation therapies in the near future. But equally, if I was given all the money in the world to work on, I would be working not only on Alzheimer's disease and these early diagnosis issues, but also on schizophrenia. Because schizophrenia is a completely debilitating condition that hits young people, the late adolescent, early adulthood. It's horrific. It destroys lives. You hardly ever get out of it. There are drugs which can reduce the psychosis, the delusions, hallucinations, but these people are left with horrendous cognitive problems. And we've recently been working on a big EU project in collaboration with industry called NewMeds, which has tried to work out new ways of developing cognitive enhancers to help schizophrenia, things like ampakines, for example. Now, this is an area which is not really well supported anymore by the drug companies because it's so difficult. But that is the kind of project that I would really like to take on board if I was given all the resources I could, because I think we could get somewhere.
I remember I read something about forms of mental illness a few years ago, something I hadn't heard at the time, but was really interesting to me, that basically the types of mental illness that are available to a person, for lack of a better term, are really mediated by the culture that they're in. People go crazy in different ways in Japan than they go crazy in the US, than they go crazy in Africa. And this is something which is sort of getting more homogenized as the world becomes more intertwined. But schizophrenia was the one exception to that. Schizophrenia is a worldwide, species-wide phenomenon that they found everywhere. And I just thought that was an interesting little asterisk next to that one.
That's a really interesting point. I mean, there have been cultural differences with schizophrenia and bipolar disorder, manic depression. They're often confused. In the UK, it used to be the case you were more likely to be diagnosed with bipolar than schizophrenia. In America, you're more likely to be diagnosed with schizophrenia. So there are these kind of ambiguities. I mean, I think that's no longer so true, to be honest. I actually think that most of these disorders are culture-free, as it were, as you say. Schizophrenia in Japan is pretty similar to schizophrenia in the US. I mean, there will be some cultural differences, but I think mental illness is a global phenomenon. And the reason is because I think the brain is basically the same. The brain goes wrong in the same way in these different cultures. And I think that's going to be shown more and more in the next period.
Is there anything else that you'd like to circle back to?
Okay, the other thing I was going to say, which you'd be interested in, is rubbing memories out. We talk about cognitive enhancement, boosting memories is a good thing, etc. There are some memories you don't want to have. So these are obviously post-traumatic stress disorder and, to some extent, addiction. So you don't want stimuli which remind you of drugs to cause craving or which then cause relapse. Recently, the last 15 years, someone called Karim Nader and Joe Ledoux, when they were working at New York University, rediscovered this amazing phenomenon called reconsolidation of memory. So I'll tell you what it is. First of all, I have to tell you what consolidation is. So for memory to be consolidated and go into long-term memory, where it goes into some kind of store and then, has a fairly permanent form. And the theory was always, once a memory gets into long-term store and it's consolidated, it's there kind of, and it may decline over time, but it's basically there. You just get it out when you need to, and there it is. It's immutable. It's a crystallized thing. What they showed is that this is a completely wrong concept. They rediscovered that if you remind someone about a memory, bring it back from long-term memory into working memory, that's conscious memory, is active. It's also labile and fragile, and you can change that memory before it goes back into long-term store. So what they showed is that that memory needs new protein synthesis to be viable. And this memory in this form, if you block glutamate receptors, which are responsible for memory, basically, the so-called NMDA receptors, not the AMPA ones, but the NMDA ones, which work in concert with AMPA to produce plasticity of neural networks. If you block those receptors during the retrieval of the memory is lost. It doesn't go back into long-term store, which is great. So in other words, it's erased. Bizarrely, they also showed that a common garden drug called propranolol, which is a beta blocker, so they're now piloting things like propranolol for use in PTSD patients, and they've made some progress. So the kind of thing you do in this experiment, for a rat, you make it frightened of an auditory sound, and then in future it shows fear for that sound whenever you present it. But if you present the auditory sound without the reason that made it fearful, which is generally a mild electric shock, and give a drug at the same time, they lose that fear memory. And it's all done by a structure in the brain called the amygdala, which is involved in emotional memories. So they've tried the same kind of thing with PTSD patients. They get them to reimagine the trauma, all those horrible memories, and at the same time they give them this propranolol, which is a safe drug, see whether that reduces their ability to remember that traumatic memory in future. And they get some effects in some clinical trials. It makes them a bit less fearful if you measure their sweating, for example, or their heart rate changes, but it doesn't do the whole job yet. So that's one of the things they're working on, the erasure of memories. And it's really interesting because, of course, psychoanalysts would say this is a bad thing because you've got to work through the memory, in order to get rid of it, And that's controversial. Do we really need to work through the bad memory? Won't we just get rid of it? So it's a very interesting area, this so-called reconsolidation of memory. And I think that's going to be a hot area in future.
Smart Drug Smarts.
So thank you so very much to Dr. Robbins for his time on that interview. That was actually one of the longer interviews that I've done recently. We went over an hour for the original talk, and a couple times spiraled off into directions that themselves I think would make worthy episodes in the future. And do not be surprised if in the semi-near future we see a Dr. Robbins follow-up episode. He's obviously a wealth of information and a man who is just passionate about the work that he's doing. It's great to hear from somebody with that level of professional credentials that, although the jury might still be out on some of the ethics involved in cognitive enhancements, the jury might still be out on some of the mechanisms of action of what's actually going on when people take these things, that at least in some cases, there's really no question as to the efficacy, that it's a measurable, provable thing, and I find that pretty awesome. Not surprising, but nice to have corroboration. But now, as promised, the Ruthless Listener Retention Gimmick.
Smart Drug Smarts. Ruthless Listener Retention Gimmick.
So as I mentioned, I'm in the midst of a water fast week right now with about 15 other Smart Drug Smarts listeners, which has been a really cool experience, by the way, getting to meet some other people in the community. If you are one of the participants and you're listening, a big hat tip to you. It's been awesome so far. Well worth the not eating food for a couple of days just to kind of meet all the people involved. I saw this on Twitter a while ago when we were first starting to talk about doing the water fast, and I figured I'd save it for later. This is a list of the effects of dehydration on the human body, and a good reminder to everybody on the importance of staying hydrated. As you've heard a million times, your body is mostly water, and of course maintaining the absolute volume of that water is actually pretty darn important. At a 1 to 3% loss in your body's water, there's no apparent change in function. However, once you reach a 3 to 4% loss, there will be decreased oxygen intake. At a greater than 6% loss of your body's water, power and strength deteriorates, and at greater than 7% loss, medical intervention is going to be required. On the flip side, there's almost no downside to having excess water in your system. Yes, you can technically overdose on water in very, very large amounts, but you pretty much have to try to do so. The rule of thumb should be to drink 1/2 your body weight in ounces per day. Now, I'm assuming they mean 1/2 your body weight in pounds. So me as a 180-pound guy, that'd be 90 ounces of water per day, which is about 3/4 of a gallon of water. Of course, since doing the water fast, I've actually lost 6 pounds in the last 2.5 days, most of which is water weight, strangely. But more on that in a future episode. There's actually a really interesting article that Rhiannon came across about how water fasting can sort of start a chain reaction that can regenerate a person's immune system. And we're going to be talking with the researchers behind that study in probably one of the next 2 to 3 episodes. So stay tuned and I'll give you all the sordid details about the water fast experience when we can get that interview lined up.
Smart Drug Smarts, the podcast so smart, we have smart in our title twice.
You heard it. That is the entire episode. If you liked what you heard, we always appreciate kind reviews on iTunes or Stitcher or other social media platforms that I may or may not even have heard of. My social network of choice is Twitter, and we have a Smart Drug Smarts account. I also have a personal Twitter account, which is @Lawlerpalooza. Feel free to ping me there. It is very likely I will be back at you at the same time, same podcast, all that stuff next week, assuming that I do not starve to death in the meantime, but I deem that very unlikely. I've got quite a ways to go before I downgrade myself into a pure skeleton, and coming up on the hump day of Waterfast week, still feeling pretty good. So thanks for listening, have a great week, and stay smart.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization. Smart Drug Smarts should be listened to for entertainment purposes only. Although some guests on the show are medical doctors, most are not, and the host is just some random guy. Nothing you hear on this podcast or read at smartdrugsmarts.com should be considered medical advice. Consult your doctor and use some damn common sense before doing anything you think might have a lasting impact on your brain.